Swayam Prabha. Digital India. Educated India.
Welcome. So we were discussing our Hodgkin-Huxley system of equations that describe the spiking behavior of neurons, and Hodgkin-Huxley type models exist for a variety of neurons. I mean, researchers have derived them for a variety of neurons based on the kind of ion channels they have, or the kind of or the density of the different types of ion channels they have, and so on. And they have stood the test of time, and actually we know a lot more today in terms of actual gating of ion channels like the sodium channel, potassium channel, and so on, as we have discussed in our ion channels lectures. And they seem to corroborate with whatever Hodgkin-Huxley came up with back in the middle of the last century. And so let us, we said that ultimately the circuit model for the neuron, remember we are doing a point model, that is a single voltage describes the entire neuron, and we have the I external that is in our control in this case, and the membrane is described by this capacitor, membrane capacitor. The different ion channels that we have, we said that this is, let's say, the G Na as a function of V. Then we have our ENA, which is the battery representing the reversal potential of sodium. That is when the voltage V in minus V out is equal to ENA, there is no net flow, that is this INA is 0. Similarly, for the potassium branch, we are assuming only one type of sodium channel and one type of potassium channel, which describes the Hodgkin-Huxley equations and is adequate to explain the spiking behavior of neurons. This is passive, the G leak and corresponding E leak, and this is our EK. So here we have, so essentially we are trying to find out what our GNA as a function of V should be and GK as a function of V should be. As we have discussed, our equations governing this system, one of the equations is C dV dt, I external minus GNA as a function of V, V minus ENA minus GK into V minus EK minus G leak into V minus E leak. So, as we said, for a clamp, we described how we can, for different voltages, by blocking one channel of the sodium and potassium channels, we can find out what the sodium current is, and that sodium current can be converted to obtaining the GNA as a function of V and T for a particular clamp. And so from this we can derive this, because we know what V and ENA are, where when we are blocking the potassium channel and the G leak and E leak are known. And I external is basically corresponding to the sodium current that is going, because we are in clamp mode, as we have discussed in the patch clamp lecture. So from this GNA, what we saw that we have two components of the sodium conductance, one that is an activation part and another that is an inactivation part. And potassium has only activation when we clamp it to a particular voltage, the potassium conductance increases and stops at a particular value. The current increases and stops at a particular value, and so the conductance also increases and stops at a particular value. And that peak value is what we will say as the steady state value of the conductance for that particular voltage. And what Hodgkin Huxley then came up with is the idea that let us represent that sodium channel gating, voltage gating, with different gates, where there are 4 gates, 3 activation gates and 1 inactivation gate. So, why exactly the numbers 3 and 1 came about? We will just briefly mention that it essentially came about empirically, and that's with the other equations that we will see. These explained the data the best, and that I mean they came up with this with certain insight, of course, which is the most important thing, and then with the numbers that fitted the data best. And now from other studies in the more than 60 years later, 50 years later, we now know that indeed there are 3 activation gates and 1 inactivation gate in sodium channels and 4 activation gates in potassium channels. So, this is for sodium channels and this is for the potassium channel, 4 activation gates. So, as we mentioned, the gates are activation gates are such that when voltage increases, that is, as voltage increases, the peak value of the conductance increases. So, this GNA peak at that particular voltage, as we saw last time. Similarly, for inactivation gate, it is the opposite, that it with increasing voltage it has to come down. So, now as we also saw that if we repeat those voltage clamp experiments, we see stochastic behavior, that is, they are not always, the ion channels, all of them are not always opening, or whatever fraction of them are supposed to open on average, they do not always open in every trial. And so, what that means is that there is a probability or a stochasticity associated with the opening and closing of these gates, or opening and closing, hence opening and closing of these ion channels. So, what Hodgkin and Huxley then proposed is that let us represent the probability of the 3 activation gates being open is represented by M. So, this is probability of sodium channel activation gate open, that is represented by M, and the inactivation gate's probability of being open is represented by H, and the activation gates of potassium channel being open, that probability is represented by N. With the assumption that these gates work independently and that they are a function of voltage, what we find is that with 3 activation gates and 1 inactivation gates, the probability of a sodium ion channel being open would be M cube H, where actually M is a function of voltage again, the cube of that, and H as a function of voltage again, and this is a sin power 1 as there is 1 inactivation gate. So, if we assume that there are a large number of gates, then on average we can say that the conductance of the sodium ion channels in the entire neuron it can be given by a fraction of the total conductance. So, if we can measure the total sodium conductance of the neuron, that is the conductance of sodium when all the sodium ion channels are open, the conductance of the ion channels when all the sodium ion channels are open. If that is GNA bar, then if it simply means that for a particular value of M and H, on average we will have the sodium conductance to be GNA bar times M cube H, where implicitly these are functions of voltage. So, this GNA bar is the overall sodium conductance of the neuron, the conductance of all the sodium channels put together, and M cube H is the probability of each of those channels being open. So, with law of large numbers, we can say that the conductance of the sodium channels on average is going to be this GNA bar and M times M cube H, again assuming independence between the different gates. And similarly, for the potassium, we have a total potassium conductance of GK bar times n to the power 4 that will describe the behavior of the conductance of the potassium channels. So, we can now rewrite this same equation that we have using these terms, the GNA bar and GK bar, and get a new equation where we have C dV dt is equal to I external minus GNA bar M cube H into V minus ENA minus GK bar n to the power 4 V minus EK minus our passive conductance G leak V minus E leak. So, now we are posed with the question, so how do M, H and N change with voltage? So, we only saw so far sort of what the steady state value of these N's may be at a particular voltage, or maybe the M's can be at the particular voltage. That is, if we allow the M gates to be held at a particular voltage, whatever is the probability of the M gates being open, that is the steady state value of M at that voltage. So, what Hodgkin and Huxley did was that the whole thing came about together, that with that equation and assuming that there's first order kinetics of the gates, they describe that for each of the gating variables, the M, N and H, these are the gating variables. They are, as we said, are the probabilities, but they are also dynamically changing based on the voltage, since they are function of voltage. So, what we have is then essentially first order kinetics dm dt is equal to m infinity v minus m by tau m v. There are a few terms here that need explanation. So, what are we saying here is that as if we hold the system at a particular voltage V for a very long period of time, so that steady state is reached. In other words, this dm dt goes to 0 in steady state, then obviously m will reach m infinity V as the dm dt goes to 0 at steady state. So, if we are holding it for a very long period of time, ultimately m has to become m infinity V. And how fast is it reaching that m infinity V is this time constant, this tau m V. And the more complicating factor here is that tau m is also a function of voltage, and m infinity is also a function of voltage, and this m infinity v is sort of the steady state value that we were referring to when we were plotting the conductance curves for, or the sodium current curves for with a voltage clamp, or the potassium n infinity v would be the potassium current curves, that peak value corresponding voltage, corresponding conductance, and obviously the associated fraction in that case, that is the probability value of, or n. So, similarly d and dt is also described in the same way, which is n infinity v minus n divided by tau n v, and dh dt equal to h infinity v minus h by tau h v. So, we have now a set of 4 variables v, m, n and h, and we have 4 differential equations governing their dynamics, that is our c dv dt equation with the I external minus our g n a bar m cube h v minus e n a, and the rest of the terms now you are completely familiar with. So, and this is the first equation here. This would be the second equation, third equation and the fourth equation. So, the dm dt, dn dt and dh dt. So, and you can see that they are all interconnected through with each other. So, m is connected to the first equation through this m cube, n is connected to the first equation with n to the power 4 here, h is connected to the first equation through this h, and each of this m, n and h are connected with voltage, because our m infinity v and tau m v and the other variables, other gating variables are all dependent on voltage, the similar equations. So, this set of differential equations cannot be solved analytically. As you can imagine, even if we first of all, the m infinity as a function of voltage, this relationship needs to be known in order to solve this system of equations. Similarly, tau m v also needs to be known in order to solve these equations, and so for the all the other gating variables. So, let us say even if we know them, still let us say we have functional forms of them, still these system of equations cannot be solved analytically and has to be simulated numerically in order to understand the behavior of the system. So, the way m infinity v, tau m v and the other ones, that is h infinity v and a tau h v, n infinity v and tau n v are obtained are with those voltage clamp experiments that we described. So, essentially with fitting the curves, Hodgkin and Huxley estimated empirically what the m infinity v function would look like with voltage, and all the others by fitting the time constants with the correct powers with the observed traces. And what they found is essentially the description is like this, that if we have a voltage in this way and this is the m infinity as a function of v, then if the resting membrane potential is around minus 60 millivolt, then there is a small value of m infinity v, and then it rises and reaches 1. So, the exact figures will be exact numbers will be given in your handouts. Here we want to understand the system simply in terms of the characteristics, and the real actual numbers will go into the simulation. So, they reach 1 at a particular voltage close to plus 20 millivolts or plus 40 millivolts. And N infinity V also has a similar shape. That is, if we have this N infinity V as a function of voltage, it is also an activation gate for potassium channels. This was also an activation gate for potassium channels, and so the steady state so potassium gate opening probability is also increasing as a function of voltage, just like the M gates. And obviously, as we have described earlier also, the N in H infinity V changes from a value of 1, and at around minus 60 millivolt it is around a 0.6 value. That is, a lot of the activation gates are open, and then it drops down and reaches 0 as voltage increases. So, Hodgkin and Huxley also came up with from this empirical data, they came up with functional forms. I mean, they are not obviously theoretically derived, but something that explained the functions that really fit the data very well, and based on those functions you can actually put those M infinity and N infinity in these equations to solve them further. Obviously, we also need the tau M V and all the other time constants as a function of voltage. And what they showed that what they found in fact is that for the taus, if we plot the tau M as a function of voltage, this is the voltage, then it is more like a bell shaped thing where it is at rest, it is somewhere at the peak value, near close to the peak value, and again the exact plots are available for you in your reading material, and essentially the nature would be something like this, that is sort of a bell shaped thing. So, this is tau H V, and this is voltage, and this is VR or minus 60 millivolts. So, notice something that they all look similar, but a very important point is the scale here. So, these values, tau M values, are very small, that is in the order of 0.2 millisecond and less, or less than half a millisecond. Tau N is of the order of 2 to 5 milliseconds, and tau H is of the order of 10 to 20 milliseconds. So, as you can see, there is from M to N to H, these gating variables, the time constant of each of the gating variables change almost an order of magnitude from M to N and then from N to H. And this essentially is the most important point in terms of describing an action potential using the Hodgkin-Huxley equations, this difference in the time constants. So, what are we saying? That the M gates or the sodium channel activation gates open extremely fast, and similarly the potassium channels are slower than the sodium channel activation gates, and so N opens slowly, and H, the activation gates, their opening and closing is further slower, almost. So, these are all in almost multiples of 5 to 10 as we go from one to the other. This as if we now put all this together, if now let us say we have the M here and we are at V rest, which is equal to minus 60 millivolts, and we have M infinity V, and we have a small sodium activation gate open and a lot of that is around 0.6 of the inactivation gates being open. So, here what we have is m is small, v equal to v rest, very small. H is of the order of 0.6. So, that means that m cube H is also going to be small, and so a very little sodium current is present at rest, which is balanced out by whatever potassium current is there at rest based on the small value of n. So, here also n is also small. So, and so basically the I n a plus I k plus I leak and I external is obviously 0, and at rest in steady state dV dt is 0. So, this is making the overall current across the membrane to be 0 at this point. And now, we saw that we have synaptic inputs which are the current injections into the neurons, or in other words, let us say that I external is we inject a small current. If we are patched onto a neuron, let us say we inject a small current in there. So, what that does is, if we inject a current I into the neuron, so let us say this is outside and this is inside, which means positive ions have gone in. That means, if this is our V in and V out, so this is the membrane, since positive ions have gone in, this V in minus V out increases. So, delta V is increasing, which is this axis here, in this curve. So, from V rest, because of the current injection, the system moves to the right slightly, let us say. So, what happens is that because of this movement to the right, this small depolarization, which is the increase in voltage, there is an increase in value of m. That is, the probability of the sodium channels being open increases from this particular value to that value. So, this is the change in the m infinity V, and since m is extremely fast, the m value reaches the m infinity V value almost immediately, instantaneously. So, what that does now is that H is starting to change, it has not changed within that less than half a millisecond period. So, H is still very close to 0.6. So, more sodium channels have opened up. Potassium channels are also slow, they also haven't moved so much, they also haven't opened so much more with this small change in voltage, because they also have a time constant of 2 to 5 milliseconds, that is they will also open, but very slowly. So, with this increase in voltage, and there is a opening of more sodium channels, and as you know that if the membrane potential is different from ENA, then the sodium, and if there is a path available for sodium to flow across the membrane, then sodium will flow in a direction such that the potential is pulled towards ENA. So, we are at minus 60 millivolts. If you remember, our ENA is at plus 40 millivolts. So, sodium will flow inwards, that is trying to pull the membrane potential towards plus 40 millivolts or this ENA. So, what that does is that increases the voltage a little further, because now positive sodium ions are going in. So, the voltage is further moved to the right. I am exaggerating this in the plot, but it can move to the right, and then so more sodium channels open, and then more sodium comes in, and this increases the voltage and more sodium channels open and goes on in a very fast time scale, less than half a millisecond or so. So, what that does is that there is a shoot up in the voltage. There is a small catch here which we will mention at the end, that if you increase the voltage, the sodium activation gates reach a saturation, that is, all of them are open, and by then the potassium channels have caught up, that is, they have started opening and there are lots of them are open, because as you saw that the time constant curves are bell shaped. So, as the voltage moves to the right, the potassium time constants are also decreasing. So, by the time all the M gates have opened, the potassium channels have all started to open, and so the potassium conductance increases, and so potassium channels being open would mean that the potassium ions will flow in such a manner that the membrane potential will be pulled towards Ek, that is the reversal potential of potassium, and so it will go towards the minus 80 millivolt. So, if we look at the voltage, what happens is that you are at V rest with that current injection, there is a sudden shoot up in the voltage and then they drop down, and by now essentially the age gates have all closed because they are the slowest ones. They are all closed, and so the sodium channels are inactivated, that is H is 0. And then briefly they, after a period of time, they will return, the entire system would return to rest. So, here it is, I have explained this very qualitatively. This needs to be simulated to fully understand it, and there is, as I said, there is something that we mean as threshold, which I did not mention while describing the behavior of action potential, as we will show that indeed there is a particular value of voltage which needs to be crossed. When the current injection is happening, that particular voltage needs to be crossed, only then there will be an action potential. Otherwise, if it is the depolarization is not large enough, it is going to come back down and not produce an action potential. So, that we will describe in later lectures, how we model that phenomena or how we understand that phenomena. So, in short, we have the Hodgkin-Huxley equations that can describe the formation of action potentials, and we will then go into how to analyze the Hodgkin-Huxley system of equations to understand the behavior of the action potential in the next lectures. Thank you.